Hello, everyone. My name is Meg Bruning. I'm the director of the Transcend program at Arizona University, which is a training program in maternal child health. Not only do we provide training for graduate students in nutrition, but we also try to provide continuing education for practitioners in the field. And we are excited to kick off our Transcend seminar series for 2020. And we are excited to have Dr. O'Brien with us presenting on myths and misconceptions of mineral metabolism during pregnancy. Dr. O'Brien is currently a professor with the College of Human Ecology at Cornell University and a true leader in the field. Prior to her work with Cornell, she received her PhD in nutrition from the University of Connecticut and then completed her postdoctoral fellowships at NIH and Baylor College of Medicine before joining the faculty in the Center of Human Nutrition at Johns Hopkins Bloomberg School of Public Health, where she worked for 10 years before moving to Cornell. Dr. O'Brien's research focuses on mineral metabolism in infants, children, and pregnant and lactating women of both developed and non-developed countries. She currently has several ongoing projects in this area. For those of you who joined a little bit early, our own Dr. Corey Wisner trained under Dr. O'Brien. So we're really glad to have those two connections. So please welcome Dr. Kimberly O'Brien.
Great. Well, thank you very much, Dr. Bruning, and it's wonderful to be here with you and with Dr. Wisner. It's a real honor to be invited to join this program. So what I want to do today is really focus, instead of broadly on mineral metabolism, I really want to focus this down onto iron. And what I'd really like to cover today is I want to briefly review aspects of iron physiology. It's the most unique mineral, I think, because humans have no physiological means of excreting it from our body once we get it into the body. And then talk about how that system is additionally challenged when women become pregnant. I would like to review how the maternal iron requirements were established and how the definition of anemia was established and what data were utilized to define anemia during pregnancy. I would then like to really discuss pregnant groups that have an increased risk of deficiency of iron and those that have an increased risk of iron excess. Both extremes can occur. And then focus on really what is unique about defining nutrient requirements during pregnancy and why is studying pregnancy so fun? Many, many reasons why studying pregnancy is exciting, but there are unique aspects to physiology during pregnancy that don't exist at any other life stage. And finally, I would like to end by capturing the other product of pregnancy, the newborn. How do we truly define a nutrient requirement if we don't see how maternal nutrition across gestation influences the status of the neonate at birth? So to begin, I want to focus on iron homeostasis. And as I mentioned, it really is quite unique because once iron gets into this system, we can't physiologically get it out of our body. And iron is also unique because of this tight regulatory necessity. We need to have all of these different tissues and organ systems communicating with one another. And to begin, one of the most important parameters to overall iron homeostasis is absorption. If it gets across the gut into our body, it's going to be there unless we bleed or slough off some iron in cells into our GI tract. So humans have evolved and other mammals have evolved a very, very tight regulatory capacity to block iron from coming into our body when we don't need it. Iron is a very common mineral in our environment, but iron is found predominantly inorganic iron in the form of ferric iron or iron 3+. And that form of iron can't be absorbed. Before humans can absorb it, it gets reduced in the gut by a ferroreductase called duodenal cytochrome B into ferrous iron, and then ferrous iron can then be taken up into the enterocyte by divalent metal transporter 1. Once iron gets into the cell can decide to utilize it for intracellular functions, to store it as ferritin, or it can export it out into the body to meet physiological iron demands. Another interesting thing I find about iron physiology is that there are multiple regulatory proteins for many aspects of iron homeostasis, but we only have one non-heme iron export protein in our body, and that's ferroportin. Non-heme iron can only exit cells through this protein. If it's exported, it crosses ferroportin as ferrous iron and then gets oxidized by hephaestin, which is an iron peroxidase enzyme. And that ferric iron then binds transferrin, and transferrin is ferried throughout the body. So the first regulatory step occurs at the enterocyte. And this step is necessary to only allow about 1.5 milligrams of iron into our bodies per day. In a non-growing adult, only a very small amount of iron is needed to maintain homeostasis. And that's because the vast majority of iron that we use every day comes from catabolism of senescent red blood cells. So once iron gets into our system, we then utilize it. The majority, 80 to 90%, goes to the bone marrow and becomes incorporated into red blood cells. So the erythroblast has the highest iron demand in a non-pregnant individual. They are loaded with transferrin receptors and they scavenge the transferrin-bound iron to make hemoglobin. Iron is incorporated into the heme molecule. And 20 milligrams of red cells every day are broken down because they're old and less flexible, and that iron is released. So iron is absorbed, most is utilized by the erythroid tissue and bone marrow, and then those red cells are constantly being recycled. So another very important cell type in iron homeostasis is the macrophage. Macrophages will engulf these old non-flexible cells, break them down, and then release that iron. And most of this catabolism occurs in the spleen, which is why you see the spleen on this graphic. And then the iron just keeps entering this cycle. If more iron is in the system than needed to maintain red blood cell mass, it becomes stored. And iron is predominantly stored when it's in excess in our liver, in our pancreas, and in our heart. So the liver is a very important site of storage. So these 4 different roles of iron in these different tissues have to have a means of communicating with one another. And the one other tissue that's integral to these pathways is the kidney. All of our blood is constantly being filtered by the kidney. So the kidney is a very unique organ with respect to its ability to sense the oxygenation level in an individual and to detect whether or not that individual is suffering from hypoxia, too little iron, and needs to make more red blood cells. So how do all these different tissues and organs communicate with one another? It's a very complicated system, and for many years, people didn't understand what were the signals that allowed the system to coordinately regulate what was going on with iron. The very first hormone that was identified was erythropoietin, and this hormone was identified in the 1950s. It's produced by the kidney in response to hypoxia. When EPO is made, it tells the red blood cells, the developing red blood cells, to proliferate to make more mature red blood cells, so the individual has a greater capacity to reoxygenate and move oxygen throughout the body. And from the 1950s, it took another almost 50 years for the next hormone in this pathway to be identified. This hormone was identified by UCLA investigators, Tom Gantz, and it's a very small peptide, which made it difficult to identify and difficult to measure. This is produced by the liver, and it also functions as an antimicrobial peptide, which is how it got its name. Once hepcidin is produced, its receptor is ferroportin. So this hormone binds to the iron export protein and prevents iron from leaving the cell or leaving the enterocyte. So it blocks iron from entering the system. It's a negative regulatory hormone. And then finally in 2014, 14 years later, Tom Gantz's group again discovered another iron regulatory hormone. This hormone was named erythroferrone, and it's produced by erythroblasts. So now people understand how hypoxia signals, iron stores, and erythroid iron demands all are regulated coordinately. What happens is EPO stimulates erythroferrone production, and erythroferrone inhibits hepcidin. If there's not much hepcidin, then ferroportin can export iron into the system. Hepcidin regulation is complex. There are many other signals that can influence hepcidin, but this is how these 3 hormones communicate. So now let's add a pregnancy to this mix. Now, if a woman becomes pregnant, she needs to start moving iron across the placenta to her fetus. And it's not a small amount of iron that this requires. At the end of gestation, a fetus has accumulated 300 milligrams of iron. And during the last 90 days of pregnancy, 5 to 8 milligrams of iron per day are deposited in the fetus. So nearly half of the iron that's released every day into our system goes to the fetus, making maternal iron demands very challenging to meet and what could be viewed as setting up a competition between the mother, the placenta, and the fetus for these limited amount of iron. We have data on erythropoietin during pregnancy, not lots of it, but it exists. We have hepcidin data during pregnancy, but we have no erythropoietin data during normal human pregnancy yet. So as an aside, we're currently funded by NICHD to look at erythropoietin in pregnant populations in collaboration with Tom Gantz and Ellen Nemeth, the individuals that discovered the other two iron regulatory hormones. And we picked women carrying multiple fetuses and pregnant adolescents because these two groups have increased iron demands. The multiple fetus population, because these women are supplying iron for multiple fetal placental units, and our population included twins, triplets, and quadruplets. And the teens have extra iron requirements because they're still growing. And they may, in essence, be competing with the fetus and the placenta for the iron that the teen needs for their own continued growth. So we will hopefully be getting erythropoietin data. We have generated data and we'll be publishing this soon. So many people have tried to estimate how much iron in total do women need to support a pregnancy. It's not just the iron that goes to the baby, but iron also gets sequestered within the placenta. Iron is needed to expand maternal red blood cell mass. Then women recover some of that iron after pregnancy when their blood volume goes back to its typical size. Altogether, it's 700 to 800 milligrams of iron over the course of pregnancy. And many women don't enter pregnancy with enough iron stores or enough dietary iron to meet this demand, and they're at risk of becoming iron deficient or anemic. I want to come back to one of these measures later with myths and misconceptions to talk about placental iron content. So I'll come back to this 90 milligram per day estimate that it's thought that the placenta accumulates over gestation. So the Institute of Medicine has tried to estimate how much iron do women need to meet this increased iron demand. And they've estimated that for each trimester of pregnancy, the first, the second, and the third. And they've assumed that absorption was 18 to 25%. You can see that by late gestation, when the fetus is actively accumulating iron, women would need to be ingesting over 20 milligrams of iron per day to maintain or meet these iron demands. The estimated average requirement in the RDA for iron were last set in 2001, 19 years ago. It's really time to update these requirements, in my opinion, for multiple reasons. One of which is at the time these recommendations were published, there was no mention of hepcidin in the DRI book, and erythropoietin had not even been identified. So we have a lot more data on how pregnant women maintain iron homeostasis. These estimations had to make a lot of assumptions, like all DRIs do. There's lots of gaps in the literature. One of the assumptions they made is that our diet has a moderate bioavailability and that heme iron comprises 10 to 15% of iron in our diet. This is one other very unique aspect of iron physiology. We don't just ingest iron as inorganic non-heme iron. Some of the iron in our diet is ingested as heme, from myoglobin and hemoglobin and animal products if you're not a vegetarian or vegan. And some of the iron is also in the form of ferritin, a storage form of iron. And each of non-heme, heme, and ferritin have unique absorptive pathways. So the Institute of Medicine assumed that a typical adult pregnant woman would consume 15% of her iron as heme and that 25% of that would be absorbed. So for a combination of all of these assumptions and factorial approaches, our prenatal supplements in the United States contain 27 milligrams of iron per day. In Canada, supplements contain 16 to 20 milligrams of iron. So if we look at the far right of this slide, we can see that the upper tolerable limit of iron is 45 milligrams per day. It is possible that pregnant women can exceed the upper tolerable limit taking this typical standard prenatal supplement. So some groups have tried to estimate if lower prenatal iron doses would be adequate, and it's why Health Canada recommends a lower dose. This modeling exercise was an exercise undertaken by Health Canada based on national data they obtained from the Canadian Community Health Survey. In that survey, they had dietary recalls in over 4,000 non-pregnant women, and they used that data to model the typical iron intake in a healthy non-pregnant woman. That's the black line at baseline. And then they added in a 16 milligram per day prenatal supplement and simulated how that curve would shift, and that's the gray dotted line. You can see that a 16 milligram per day iron supplement moves the majority of the population over the estimated average requirement, with only a few percentage of women falling below the EAR. If they increase that from 16 to 20, all women now fall over the estimated average requirement. If they increase that even further to 27, which they didn't show on this figure, 33% of the women would be ingesting over the upper tolerable limit. So they recommend a slightly lower prenatal iron dose than the United States. This is a wonderful exercise and something very useful to do to see what prenatal mineral intake supplement does to the typical intakes, but for iron, there are some unique aspects to this. And that goes back to the point that all iron in our diet is not in the same form. We consume the non-heme iron and heme iron. Remember, I mentioned that the DRI estimated heme absorption at 25%. There were no data on heme iron absorption in pregnant women. To address that gap, we intrinsically labeled a piglet. We took a piglet at birth. We injected the piglet with 150 milligrams of stable iron isotope. We then sacrificed the piglet and made pork chili and fed intrinsically labeled heme iron to pregnant women and non-pregnant women, and found that 50% of the heme iron was absorbed. So double the amount that was estimated by the Institute of Medicine. Of greater interest is that heme iron absorption was not regulated in response to a woman's iron status. Both pregnant and non-pregnant women absorbed 50% of the heme iron. If at the same time we gave the labeled heme iron, we fed these women another stable isotope as ferrous sulfate, the form that is often tested to look at non-heme iron absorption. And in that study, we did find that non-heme iron absorption was significantly higher in the pregnant woman compared to the non-pregnant woman. So heme absorption isn't impacted by hepcidin. So a woman doesn't downregulate it when she's iron replete. This is just leads to perhaps a concern that a large amount of supplemental iron in a woman that's taking a lot of heme iron in her diet may not be necessary. So it's important to consider both the quantity of iron a pregnant woman ingests and the composition of iron that she has in her diet when you're looking at the exposures to dietary iron across pregnancy. And variability in this measure may lead to a lot of noise in data. If you look at supplementation studies looking at responses to iron intake, they very often do not adjust for the type of iron that that pregnant individual is ingesting. And they don't break the total iron of the diet down into the heme and the non-heme components. We do know that pregnant women are able to regulate non-heme iron absorption in response to their status. But because we can't get rid of iron once it gets into our body, we don't markedly increase that. For every 10 microgram per liter decrease in serum ferritin, our work with stable iron isotopes has found that iron absorption only increases 3% for every 10 microgram per liter decrease in stores. So we can't ramp it up extremely high, and it will take women some time to regain or recover the iron they might need. Another important thing to consider in these studies is that the woman's iron status at baseline is going to influence her response to supplementation. Quite often when a supplementation study is evaluated, the baseline iron status of the individual is not taken into account. And because you absorb more iron when you're iron insufficient, it's important to control for the baseline iron status of the woman at the start of supplementation. Another interesting component of iron physiology during pregnancy is many pregnant women are, when they become anemic, are told to take 40 to 200 milligrams of iron, very large doses of iron. But we know that as the load of iron presented to the gut increases, the percent you absorb decreases. And what that ends up doing is leaving a very large amount of unabsorbed iron in the gastrointestinal tract. And at present, we don't know how that large amount of unabsorbed inorganic iron affects the gut microbiome. And that may lead to dysbiotic changes in the gut microbiome. And it may also give that pregnant individual more side effects. One of the adverse consequences to iron supplementation is that it gives some women constipation, it can cause your stools to be dark and cause nausea. So there's a lot of issues with prenatal iron supplementation that need to be interpreted and considered when recommending doses. So iron has a very unique physiology, lots of hormones involved in regulating it, and a lot of nuances in supplementation patterns. Anemia, when we don't have enough red blood cells or hemoglobin in our body, is very prevalent. This is the WHO map that they published in 2015 to show you the prevalence of anemia across pregnancy. You can see the darker areas are areas of the world where severe anemia, more than 40% of pregnant women have anemia during pregnancy. So this remains a very common problem using the current definitions of anemia. If we focus on North America, you can see that according to this map, on average, only mild anemia, the prevalence of anemia is mild in North America. And I want to come back to that point later in the talk. So the thresholds, how do we define anemia? Anemia is defined as a hemoglobin less than 11 grams per deciliter. And it sometimes is adjusted for trimester because you have plasma volume expansion. The cutoff in the second trimester is lower than the cutoff in the first and the third. Some groups recommend decreasing the threshold for Black individuals because the hemoglobin distribution seems to be shifted to the left in Black adults. But this cutoff of 11 is widely utilized throughout the literature. It's advocated by the CDC. It's used by obstetrical congresses and consensus building organizations and by the WHO. So I want to draw your attention to this workshop that was held in 2017. If any of you would like to learn more about iron homeostasis in women and young children, this is a really nice resource that NIH and the Office of Dietary Supplements convened a workshop to really review the existing literature on universal prenatal iron supplementation and to really think deeply about whether or not this supplementation had benefits and if it had risks. So there's a lot of information in this supplemental volume for those of you that are interested in learning more about iron in vulnerable population groups. But for this conference, I was asked to summarize iron status based on longitudinal data in North American pregnant populations. And I naively assumed that there was probably a large amount of data on this topic. And one of the first things that I did in reviewing that is to think a little bit more about how the definition of anemia came about. What longitudinal data had been utilized to set the cutoffs that are used globally to define anemia? So I went back to these original papers. This is the original data from the Centers of Disease Control, the table that they published looking at mean hemoglobins in a group of pregnant women at various stages of gestation with longitudinal study designs. And the little footnote to this is that these data were based on pooled data from 4 European surveys of healthy pregnant women taking iron supplements. And you can see what they did then is they looked at the 5th percentile, and the 5th percentile value at 12 weeks, 24 weeks, and 32 weeks formed the basis of these cutoffs. So I was curious about the composition of this cohorts that were used for these standards. And what I discovered is that these 4 early papers were undertaken all together in less than 400 women. All 4 of those studies did not reach 400 individuals. The racial composition of this reference population was not mentioned, but these studies were undertaken in the Finland, Sweden, and the United Kingdom. And one can assume that they were likely not representative of the current minority composition in North America. And that's a concern because CDC has recommended adjusting the standards to the left among African Americans, and the composition of the reference population wasn't consistent. The pre-pregnancy BMI was only specified in 2 of those 4 studies, and in those 2 studies, it ranged from 18 to 21. That's not representative of the current North American population, where many women enter pregnancy overweight or obese and gain excessive amounts of weight across gestation. Because obesity increases systemic inflammation, and inflammation stimulates hepcidin production, there's a concern that individuals with excess adiposity will have high hepcidin and be at greater risk for iron insufficiency. So pre-pregnancy BMI is an important factor to consider when setting references. And then finally, those 4 population groups were in supplemented women. Those women were receiving between 65 and 200 milligrams of iron per day, up to 7 times higher than our current RDA. So this is an example of going back to the original literature and really informing yourself on where kind of reference cutoffs, how they were established. So back to summarizing the longitudinal data, that's how the cutoffs were set. The U.S. Preventive Services Task Force found that there were 12 longitudinal iron supplementation trials that had compared iron supplementation to no supplementation in pregnant women. Only 3 of those studies were done in North America, and they were disproportionately done in at-risk populations, so lower income minority populations. The United States does have cross-sectional data on iron status in pregnant women across gestation, and NHANES data collects data on hemoglobin, serum ferritin, transferrin receptor, and total body iron. So we do have some normative data in our country on what typical iron stores are at various trimesters of pregnancy in US women. When I tried to find these data in Canada, I discovered that the Canadian national cohorts, the cross-sectional cohort studies, hardly recruit any pregnant women. And the last one, the one in 2007 to 2008, only had 38 pregnant women out of over 5,000 women. So they don't even try and break down status by pregnancy. So this just really emphasizes that no longitudinal national data are obtained, and the cross-sectional data are limited. Even in our country, we don't have large numbers of data. So where do we have any more larger cohorts that we can look at those cutoffs that were established in less than 400 women? This paper was published in The Lancet in 2013. This study recruited 257 population studies from 107 countries and pooled these data to look at the distribution of hemoglobin in pregnant women. This is the red curve, and in non-pregnant women is the blue curve. The 2 dotted lines are the hemoglobin cutoff to define anemia by the WHO cutoff. The red line is for pregnant women, the blue line in non-pregnant women. So the first thing that you can notice is that the mean level is close to 12. And if you look at 11, you can see everything to the left of that dotted line would be defined as anemic. However, that 11 was the 5th percentile in the other study cohort. The 5th percentile in this distribution looks like it's well below 11. So we really, I think, need to pay more attention on the current hemoglobin distributions in pregnant women from North America when establishing cutoffs to define anemia. And these hemoglobin thresholds do have some concerns. You have probably seen many times throughout the literature that it's assumed that half of the anemia in North America is due to iron deficiency. Yet most studies don't report and break down the iron deficiency anemia and iron deficiency anemia percentages. And when these data are reported, typically only a small amount of the anemia can be classified as iron deficiency anemia. And why might that be? It may be that the hemoglobin cutoffs used to define anemia are inaccurate. It may be that our cutoffs that we use to define iron deficiency are also inaccurate. At present, those cutoffs are not adjusted for stage of gestation. And at least 2 of our iron status biomarkers we know are acute phase proteins, and they increase when you have inflammation going on. And pregnancy is a condition associated with a lot of inflammation. So more data are needed on iron status biomarkers as a function of inflammatory status and stage of gestation. And there are people working on that question. It may also be that in otherwise well-nourished North American populations, there may be other nutrients that are contributing to the anemia. And an example I can give you from our studies in otherwise well-nourished pregnant population. So our pregnant adolescents often enter pregnancy overweight or obese. They gain excessive amounts of gestational weight gain on average. And we found that the anemia in our adolescents was 4 to 8 times higher in teens that had low vitamin D status. So teens whose 25-hydroxyvitamin D status was under 20 nanograms per mil had 4 to 8 times greater odds of anemia. So other nutrient deficiencies are likely contributing to anemia in otherwise what one would classify not weight deprived populations. So I'd like to come back to this global map. This global map of North America shows very little anemia in pregnant women. So if we look at our national data right now, NHANES data has identified what are some of the risk factors for risk of iron deficiency and anemia in pregnant women in our country. Women have a greater risk of iron deficiency and anemia in late trimester pregnancy. That's when the fetus is growing the most rapidly, more iron is being shunted to the developing fetus. Women that have a parity of 2 or more also have a greater risk of iron deficiency. And NHANES data have also found that the risk of iron deficiency and anemia is greater among minority pregnant women. So greatest in non-Hispanic Black, over Hispanic, over non-Hispanic white. In our research studies in the women carrying multiple fetuses, these were women again carrying twins, triplets, or quadruplets, almost 40% of women had depleted their iron stores at mid-pregnancy, and over a third of the women were anemic at delivery. And keep in mind that women carrying multiple fetuses, our cohort, none of them delivered past 37 weeks. They delivered on average at 35 weeks, and over a third were anemic at 35 weeks. This was an increase of 3-fold over pregnancy. Another vulnerable group is pregnant adolescents. In our studies in pregnant teens in Rochester, 22% are iron deficient at mid-gestation, and again, a third were anemic at delivery. Similar to the multiples, it was a 3-fold increase. Some populations of pregnant teens may have even greater risk. When I was working at Johns Hopkins, we went through all of the medical charts in pregnant adolescents that had received care there for a 10-year period at their adolescent pregnancy clinic. So in over 1,100 adolescents, 66% of the teens were anemic at delivery. So even in the United States and otherwise not weight-deprived populations, there may be a large, in certain subpopulations that have a high risk of anemia. And then it's important to just keep in mind that we need to pay more attention to the impact of obesity and inflammation on iron status. So I have focused so far on deficiency, but I indicated that I'd also tell you about the other part of the spectrum. What about too much iron? So you've probably all heard of hereditary hemochromatosis, a genetic disorder that's more common in Northern Europeans. or hepcidin is not appropriately regulated, so you keep absorbing more iron than your body needs, and you can develop iron overload. This study, the HARE study, was undertaken, and it's called the Hemochromatosis and Iron Overload Screening Study. They recruited almost 100,000 individuals from North America, from US and Canada, and they broke them up according to their race or ethnic group. And they looked to see whether or not, what was their ferritin concentration, so how much storage iron did these individuals have, how many had excess transferrin saturation, and how many had excess iron stores and excessive transferrin saturation. And what they discovered is the group at highest risk for iron overload wasn't the Northern Europeans that suffer from hereditary hemochromatosis, it was individuals of Asian descent. This drew a lot of attention to be concerned perhaps that there are subpopulations of Asians that may have a higher risk of excess body iron stores. And this may be why Asians are exhibiting a higher risk of metabolic diseases like diabetes at a lower body mass index. Remember, when excess iron is present, it is deposited in your liver, in your spleen, in your pancreas. It can cause diabetes, it can cause hepatic problems, it can cause cardiomyopathies. So excess iron stores were evident in Asian individuals. Among these 100,000 individuals, they tried to evaluate risk of excess iron stores in females in this study. So from the HARES cohort, there were 20,000 women that were not pregnant or not breastfeeding, and almost 2,000 women in that cohort were either pregnant or breastfeeding. Looking at just that subpopulation, they found there was an increased risk of iron deficiency among non-pregnant and non-breastfeeding Hispanic and African American women, similar to the NHANES data. They found that Asian American ethnicity was associated with a decreased risk of iron deficiency in all of the women. And they also found that Asian American ethnicity was associated with significantly increased iron stores in their non-pregnant, non-breastfeeding women, and that was their larger sample size. So this does draw attention to the need to be aware of possible concerns with universal prenatal iron supplementation during pregnancy, particularly in some ethnic groups, and Asian Americans in particular, if they have a predisposition to absorb more iron than they need, may be accumulating excess amounts over pregnancy. We currently have funding from NIH to look at the genetics of iron absorption, because that's where we regulate iron status. We're recruiting a group of 500 individuals, half are Caucasian, half are Asian, and they're non-pregnant, men and women, and we're going to look at iron absorption and try and identify genes associated with iron absorption, hopefully with a long-term goal of applying this to pregnancy. I would like to move on now. I've talked about the woman and what happens to the pregnant woman, but we all know that during pregnancy, there are two other things that we really need to consider. One is, does the placenta have enough iron to maintain its metabolic functions? And the other is, does that pregnant woman have enough iron available to fully endow her newborn with as much iron as it should have at birth? The chart I showed you earlier assumed the placenta has 90 milligrams. The 300 milligram per value for a newborn comes from very early studies by Elsie Widdowson, who chemically ashed human newborns to come up with this estimate. And those studies are not likely ever going to be undertaken again, so we're not going to get a lot more data on that question. But we can get our hands on the placenta. And this really made us wonder, where did this 90 milligram per day value come from? We have freezers full of placentas in our lab from the women that have done our studies. And we started to think back and figure out, where did that value come from? And it appears to come from this early paper from 1961. This was a study that recruited 50 women, so another very small study. These 50 women, you can see, are incredibly diverse. I think you could not find a more variable group of 50 pregnant women if you really worked to do this. These women ranged from 15 to 42 years of age. They'd had from 0 to 9 babies. They'd lost 24 pounds up to gaining 57 pounds. Hemoglobins from 8 to 14. Baby weight from extremely very low birth weight, 1.5 to 4.2 kilograms. And some of these women were carrying twins. So highly diverse population to set a normative value. These included low-income women, pregnant teens, women carrying multiples. It was a relatively small sample size, and it was 1960, so the methodology they used was colorimetric, which would now be outdated. They also didn't specify where they sampled tissue from the placenta, and this value really hasn't been questioned or re-examined for 60 years. It's another one of these examples where something gets into the literature and it becomes fact. And all too often, it's not appropriately reevaluated. So one of my current graduate students, Alexa Barad, is undertaking a systematic review of the literature. She has pulled out all papers that have published data on placental iron content. And this table here just reflects some of the more recent data on placental iron content using state-of-the-art quantification techniques. The red values are our data in pregnant teens. We also have data in the multiples. And you can see that it looks like the placenta contains more like 50 milligrams of iron, not 90. So maybe almost half of what those early estimations had assumed. And again, this may not make a huge thing, so 50 milligrams versus 90 out of 800, but it's about 10% is what the original value was, and now it may be 50. So it's important to reevaluate some of these dogmas in the literature. One other example I'd like to share with you is when I started in this field, I read a lot of papers that would refer to the human fetus as the perfect parasite. And the concept was that this perfect little fetal parasite would accumulate whatever it needed, and the maternal nutritional status didn't matter or wouldn't matter. When you think about that, it doesn't make any sense. It doesn't seem logical. A human fetus, when it's born, is not going to be able to survive without the mother. And if the mother were to become anemic enough, she can have an increased risk of mortality from severe anemia. So going back to this partitioning issue, can the fetus control what it gets from the mother? Does the mother control what gets passed across the placenta? And can the placenta do some control of this process? We do know that a newborn it begins to make, a fetus makes hepcidin, erythrophorone, and erythropoietin in utero, and yet that concentrations of these hormones in the baby are independent of the mom. So in our multiple birth model, we can look at one maternal environment and 2, 3, or 4 babies and start to see how variable hormone concentrations are in these newborn siblings. And babies make their own hormones in response to the iron they're getting across the placenta. To answer the question of whether or not the baby is getting enough from the mother, we really need to know what is optimal. What should a human newborn contain at birth? We're not going to be able to do those early studies of chemically ashing. We have to rely on iron status biomarkers and measuring them at birth before all of the very rapid changes occur over the first few weeks of birth. So looking at this, it started getting us thinking about interpreting iron status biomarkers at birth. Sorry, my phone, I'm going to hang it up here. Sorry about that. All right, so what are the iron status, the norms in a newborn? And any of you that have had babies know that you don't want anyone to come take blood out of your newborn unless it's absolutely necessary. But fortunately, you can get blood out of the umbilical cord. You can get cord blood, and you can begin to evaluate what are the iron status biomarkers at birth. So far, when people are looking at iron status biomarkers in a newborn, they are interpreting them in exactly the same way they would interpret them in an adult. They're assuming that a newborn is a small adult, which we know for iron is not true. And we know that that's not true because think about what's happening in utero. That fetus in utero needs to prioritize iron to make hemoglobin. It needs to be able to scavenge oxygen across the placenta from maternal circulation. A baby is born with very high hemoglobin concentrations compared to what an adult woman has. The baby has higher hemoglobin concentrations. The baby also has a unique form of hemoglobin that has a greater affinity for oxygen. Then at birth, what happens is that baby goes from a hypoxic intrauterine environment to a normoxic environment. And all of a sudden, all of the hemoglobin that neonate has is no longer necessary to scavenge iron. So a newborn catabolizes that hemoglobin and redistributes the iron that's released into stores because that iron can't be excreted. So there's a lot of very rapid changes that occur in iron biomarkers over the first few weeks of life. And there's not a lot of data in a healthy baby over that time span to let us know how the iron is redistributed. At present, the literature really looks at iron adequacy in a baby by its ferritin stores, assuming the more ferritin the baby has, the better its iron stores. But babies disproportionately put iron into hemoglobin. So is that an appropriate assumption? In our studies in newborns, we did not see that. We found a significant inverse association between hemoglobin and ferritin. The more hemoglobin the baby had at birth, and remember hemoglobin is a big iron depot, the biggest iron depot in a newborn, the less ferritin it has. From autopsy data, we also know that a fetus will prioritize iron for hemoglobin even at the sake of other organs like brain iron. So in all populations that we studied, our teens and our multiples, there was this significant inverse association. This prompted us to review the literature on cord serum ferritin concentrations and hemoglobin. So another grad student from the lab, Katie Delaney, did a systematic review of this. She found 106 articles that had presented hemoglobin and serum ferritin in cord blood, and then she classified them according to their findings. Most didn't present any correlation between those two biomarkers, and I think they didn't talk about it because it didn't fit the scenario, the assumption that people had made. That's my assumption. Only 3 studies found a positive association between hemoglobin and ferritin like you would see in adult. 7 didn't find any association, and 6 found a significant inverse association. So a far larger proportion of this population found the opposite to what has been expected in the literature. And I'm raising this just to really get individuals to question assumptions and really to highlight the need to obtain normative data on iron status in a newborn. So I want to just summarize by saying some of the things that we really can tell on iron homeostasis during pregnancy. There's a lot of evidence to support the benefit of maternal iron supplementation with respect to its ability to increase maternal hemoglobin concentrations and neonatal hemoglobin concentrations. But there isn't enough data at present to really fully know what the risks and benefits of prenatal iron supplementation are, and more data are needed on this question. We also should be evaluating the optimal iron supplementation doses. And this is true if we use our current definitions of iron deficiency. 30 to 40% of North American women are iron deficient by late pregnancy. The large cohort studies I talked about that are ongoing in Canada are finding even in Canadian well-nourished women, many women have depleted iron reserves by the end of pregnancy on current supplementation, with current supplementation. I mentioned the hemoglobin cutoffs and believe that we need more data to really evaluate these thresholds and to determine if these thresholds are accurate for current North American women. In addition, to look at our thresholds and definitions of iron deficiency with the iron status biomarkers that we have. We do know that there are race and ethnic differences in the risk of iron deficiency and iron excess, and perhaps we need to better evaluate these curves as a function of race, ethnicity, as well as identifying what are some of the genetic factors that influence iron homeostasis and humans' ability to absorb iron. Newborns, as I mentioned, are not small adults. We need better normative data on what a healthy newborn should have in its body at birth with respect to iron stores, and better try to define how that changes over early infancy. Right now, the American Academy of Pediatrics doesn't recommend screening for iron status until baby is 12 months of age unless they have risk factors. We don't know if there are cohorts or populations of newborns that have an increased risk and should be screened earlier. And if we were to screen them earlier, we need better norms so that we can identify who is insufficient earlier on in life. And the final function that I think is very important to think about is it's not just the woman during pregnancy, it's not just her newborn at birth, but growing appreciation for the role of the placenta and how the placenta functions to respond to maternal and fetal status and how it regulates moving nutrients to the baby across pregnancy. Because this is a training session, I'm just going to share with you these lessons that I have learned and some of these examples I've shown and shared with you today. It's important to challenge assumptions. I really didn't appropriately challenge many of the assumptions and things that I had heard in the dogma and the literature, and it's important to do that. I really strongly believe and increasingly believe this, that it's important to go back to the early literature and determine how many of these facts came into presence and see where the gaps in the literature are, and not just blindly accept dogma, but really evaluate the level of evidence that was utilized to develop these facts. And as trainees in maternal and child health, you can really make a substantial contribution to this field. It's such an important area. It's such a fun population to work with, and I wish you all the best in your studies. So I'll stop there, and I'm happy to answer any questions if you have any questions.
Thank you so much, Dr. O'Brien. This was fabulous. I hope you feel the applause coming from our silent attendees. We do have one question, and so this is my first time using this technology, everyone. I think you can raise your hand, and then I can unmute you if you want to ask the question yourself, but I'll read the question that was typed in. And this comes from Victoria, and she asked, I've seen articles suggesting expecting mothers to not clamp or cut the umbilical cord immediately and wait for 1 minute after birth, claiming to help newborns have more hemoglobin and iron levels. Is there really strong data to suggest this is true?
Yeah, so that is such a good question, Victoria. I was talking to a class just a week or so ago and talking about this, and the student asked exactly the same question, that I should start maybe talking more about that during these talks. But you're absolutely correct that if you delay the cord clamping more, as a baby is born, that cord pulses, and many care providers wait until the umbilical cord stops pulsing before they clamp it, with the goal of getting more of that blood into the baby. Other approaches are to hold the baby below the level of the placenta so more of that blood can drain into the placenta. For the pregnant adolescent population that we work with, midwives run those clinics, and midwives are typically very strongly passionate about delaying cord clamping. So if you delay cord clamping, there's lots of data to show that that does increase the amount of blood that gets into the baby. There are systematic reviews that have looked at what are the benefits and side effects or possible adverse effects of that. So it has been found that delayed cord clamping does increase hemoglobin concentrations in a newborn. So it's something that's very important to consider doing, especially if you're working with a higher risk population. On the flip side, what's the disadvantage? The disadvantage is hemo concentration. So you have a greater risk of jaundice if you delay cord clamping. So remember, as you're catabolizing red blood cells, you're releasing the pigments, and the baby has to deal with those metabolites of hemoglobin catabolism, those pigments. So an increased risk of jaundice, but you can treat jaundice. So that's the one disadvantage they found to delayed cord clamping. But an excellent question, and something that is easily applied and can be done anywhere. You don't need anything other than a stopwatch or an eyeball or waiting before you clamp that cord.
There aren't any more questions coming in yet, so I'll ask one. So in my class, we recently talked about the Dietary Guidelines Committee's report for infants and young children, and it's the first time that they've really looked at that, as well as pregnancy. So can you, if you're aware, I don't know if this is beyond, but can you talk about that report and your feelings on their findings?
Yeah, so I was on one of the technical expert committees for that birth to 24 and kind of reviewing the data. I think that it's so exciting that they're going to consider birth to 24 months. All too often in control data or reference populations, you start looking at something far later in life, and it's those early time periods that are so important, and that pregnancy part is so important. So I think the attention to a birth to 24 period and trying to acquire normative data over that time interval is a really admirable goal. And as that attention is focused on those key ages, more data are going to come in. For the group that I was working on looking at breastfeeding and health outcomes, it's very hard to interpret the data that's out there, because even trying to classify how is a baby fed, there's often not enough data in papers. You know, when people say breastfed, you don't know if it's breastfed with other complementary foods. You don't know if it's breastfed and formula fed. You don't know if it's donor milk or exactly what that baby is getting. So I think as the attention is focused on these age periods, there's going to be better standards of data collection and better ways of collecting that data so you can really more fully answer the questions that are out there. So definitely, I think no one would argue that breastfeeding isn't best for multiple reasons, but there's a lot of literature growing to kind of identify where are the gaps and what are the things that we need to be most concerned about.
Sure. I mean, most things, it seemed like there was insufficient evidence for the committee to feel as though they could report on it.
Yeah.
Are there any other questions from the attendees? And I'm not seeing any hands raised, so I apologize if you're raising your hand and I'm just bad at the technology. Okay, I'm not seeing anything. So thank you again, Dr. O'Brien, for your time and your expertise. We really appreciate it. For the attendees, our next Transcend seminar is on October 1st, and that is Dr. Angela Odoms-Young from the University of Illinois at Chicago, who will be talking about addressing social and structural determinants of dietary behavior in the U.S. using community-based participatory approaches to advance health equity and food justice. So a bit of a shift from what we talked about today, but just as interesting. And thank you again for your time.
Thank you. Thank you for the invitation.
Bye.
Bye.